Good morning. I'd like to thank you, the organizers, for inviting me to this meeting, um, which in which I will actually talk about bioinformatics. And let me start with a slide that I found actually quite striking. This is just the result of a search in Google of a term made by a prefix and the suffix informatics. And I think that, as you can easily see, the number of terms which contain the word bioinformatics is orders of magnitude larger than the number of terms containing any other combination of a prefix and the suffix informatics. Um, and I mean, I don't think that this is because in biology we are actually generating more data than in astronomy. And actually, I do not know the reason of why the success in Google of bioinformatics, but actually during my talk, I will try to suggest some possibilities that would explain this success again of bioinformatics. And it is always difficult to pinpoint when a particular discipline started. But I think that the roots of bioinformatics can be actually traced back to the work of this woman, Margaret Dehoff. What she did in the mid-60s actually was something very simple. She collected the sequences of amino acids of all proteins that were known at that point. As you may know, proteins are polymeric molecules, which means that they are made of the repetition of a limited number of basic elements, right? These are, in this particular case, 20 different amino acids, each of which is represented by a letter. So this is the sequence of a particular protein, the sequence of amino acids of a particular protein, the human growth hormone. The other major biological molecule is the DNA or the RNA. In this case, this molecule is made of the repetition of 4 basic units, the nucleotides, which we represented by the letter ACGT, very globally, very generically. The sequence of nucleotides of the DNA actually determines the sequence of amino acids of the proteins through the genetic code. So what actually the compilation of many sequences in the same site allowed for was for the comparison of the sequences. As you know, when we compare things, we can extract generalities from the particulars of each individual instance. And what they have built was these things called sequence alignments. These are just arrangements, matricial arrangements of amino acid sequences. In this particular case, this is the sequence of, this is a fragment of the sequence of this protein here in different species from all domains of life, animals, plants, bacteria, yeast, and archaea. And actually, each column corresponds to what's called an homologous amino acid. So this hypothetically this position corresponds to the ancestral position of the organism from which all these organisms originated corresponds to, right, belongs to, right? So this is this position. And what we can see actually is that some of these positions are extraordinarily conserved across billions of years, right? These positions are always the same. Some other positions are less conserved. Some other positions are actually much more variable, and some positions are even absent in some species, right? Or in some other species. And what, of course, what Margaret they have observed is that the species that were closer phylogenetically, for instance, animals, had a much more similar sequence than sequences that were much distant phylogenetically, like instance, an animal and an archaea. And of course, the assumption was that these positions that were more conserved and other positions were more important for the biological function of this protein. But what actually Margaret Dehoff did was, by looking at alignments of these in many different protein sequences, is just to compute how often did she see 2 different amino acids in the same column, in the same position of the alignment. And she built this thing which is called substitution matrices, which I will not go into the details, but this is simply the likelihood of observing 2 different amino acids into the same position in one of these in the same column, in one of these across many multiple sequence alignments. So that, and this is that was actually quite important, because this allowed for an objective criteria to build alignments, right? To build alignments, to allow for these alignments to be built automatically, given 2 sequences. For instance, these 2 sequences here of amino acids, which apparently are not very much related. So now we had an objective criteria to build what is called the optimal alignment. So the alignment that reproduces best that we have seen so far in the alignments that we have built, and that hopefully recapitulates which had happened during evolutionary history. Because now the optimal alignment of all possible alignments between two amino acid sequences is that alignment that maximizes the likelihood of the aligned amino acids at each position. Because we had, and that's so because we had this objective criteria, now we could actually build automatically alignments between two sequences or even more sequences. The problem, right, is that the number of alignments, even for between two sequences of moderate length, let's say 100 amino acids, which is very short, it's enormous, 10 to the 200, which is cannot be computed using the computers that we have today. So this is why during the 70s, methods were invented to make this problem tractable. And using dynamic programming, methods were invented that were very efficient in building these optimal alignments that were actually linear with the length of the sequences. This was the work first of Needleman and Bunch, theoretically, and then for practical applications, the work of Smith and Waterman in the early 80s. And I could say that with this, right, with the invention of algorithms that were actually devoted to address a biological problem, of computational methods which were addressed to solve a particular biological problem, that bioinformatics was born, right? And I just wanted to mention something, to stress something here. This data is not strictly quantitative. In science, we are used to work with numbers, with quantities, with real numbers, right? These are symbolic. These are characters, right? And this is one of the peculiarities, right, of the biological data. That some of the biological data that's being generated that has to do with the biological molecules is symbolic in nature, and therefore it's very amenable to computational work. So during the 70s, also methods for DNA sequencing were invented, and these methods were rapidly employed in many labs around the world. And in the early 80s, the first electronic databases were set up. In the United States, the GenBank was set up in Los Alamos National Laboratory in New Mexico, where maybe some of you know the Manhattan Project was developed during the Second World War. The number of sequences grow immediately very, very rapidly, sort of in an exponential fashion. And methods need to be invented to accelerate the comparison of sequences. So these HASH methods and more sophisticated methods, so that it was possible to, given a sequence, compare the sequences against all the sequences that were already known before and were stored in these electronic databases. Because when you find similarity between two sequences, you can infer that these two sequences correspond to proteins that have the same function. And actually, using these database searches, was done Doolittle in '82, found this relationship based on the similarity of the sequencing between oncogenes that are genes that are involved in cancer processes and growth factors. And this is an alignment of an oncogene and a growth factor. And the row in the middle just indicates which amino acids are identical between these two sequences. And this finding actually illuminates the molecular basis of cancer, that, as you know, is an abnormal growth of cells. These and many other findings actually already pointed out, underlined the importance of computation in biology in general. And this is a list of the most cited papers in biology during the decades from the 80s to 2000, right? And one of the most useful, one of the most used, one of the most cited papers in the field of biology during this period, during these 20 years, was actually a method that described a program to compare sequences. It's called BLAST, which already suggested that computation was very important in biology. But actually, what the sequencing methods brought up to was the capacity to sequence genomes. This is a fragment of the human genome sequence. As you know, the genome sequence encodes the instructions that determine the traits of organisms. In my case, the color of my eyes, my skin, heart, etc., etc. They actually encode the differences between the species and encode the differences between individuals within the same species. But the encoding is actually restricted to small fractions of the genome, which are called genes. Actually, in the case of the human genome, the genes correspond to less than 2% of the entire human genome sequence. And one problem, a computational problem, is to identify where in the sequence of the human genome are the genes that actually are the responsible of the biological traits. The genes have actually particular sequence compositions that make them potentially identifiable by computational means, by essentially pattern search. And this is just, I will not go into the details, but this is actually a position weight matrix that defines one of the sites that's involved in the definition of genes. Essentially, a position weight matrix is simply a matrix of the probabilities of finding each of the 4 nucleotides in the different positions of the motifs, right? This is a visual representation of this, which in this case are called splicing sites. So these are sites that are involved in the definition of the genes. And as you can see, around these sites, there is a bias in the composition of the sequence. So many methods were during the 80s, the 90s, actually early 2000s, using everything like neural networks, hidden Markov models, support vector machines, Bayesian networks, logical models, classical statistical multivariate methods to identify these and other sites that could be functionally relevant into the sequence of the human genome. And actually, when the human genome started, it was obvious that without, as it says, this is the document from the Department of Energy that actually launched the Human Genome Project in the United States. Within powerful computers, genome research cannot proceed. So the Human Genome Project took place during a little bit more than a decade. And the drafts of the human genome sequence were published early in the century. So it will be 20 years next year, by the way, in a few months from now. And of course, the growth of the data generated by molecular biology, mostly by the sequence instruments, continue to grow exponentially, or even more if you want, right? This is the growth of the number. This is, I don't know, this is the number of human genomes, but doesn't really matter. You could put the number of sequences, the number of nucleotides, essentially, and actually the growth, it duplicates more than essentially every year, right? So this would be the equivalent of the Moore's law. So this grows much faster than the Moore's law. And I actually wanted to say that you may actually think that this is something that just happened because of our technological advances, but actually I would say that you need to understand, I think we all need to understand that biology is, compared to other hard sciences, a discipline which is based on the collection of data. But this is not new, right? This is from my colleague at the CRG Center in Notre Dame. This is the number of scientific expeditions that took place during the 18th and the 19th centuries. And these expeditions were already data collection expeditions. And you can see this in the natural history museums, where actually you see only a small fraction of all the data that had been collected, and that mostly remains actually still unexplored, right? So I think that what I just wanted to point out, that this large data production nature of biology is something that will continue during the next years and in the next decades. And actually this paper already pointed out that genomic data will surpass astronomical data in terms of volume of data generation by 2025, right? I would like to just emphasize a few areas in which large amounts of data that will make the data that we have generated so far look minuscule will happen during the next years. And these are three different areas. One is in personalized medicine. I mean, the sequence of the first human genome took about 15 years, cost $3 billion. It involved hundreds of scientists. Now we have projects that can generate, we have machines that can generate hundreds of genomes per week, or at least per month. And there are projects already worldwide which are aimed to obtain the sequence of each individual on Earth. This is the UK project, which they have already sequenced 100,000 individuals. But the important thing is not the size of the human genome sequence, which actually, by the way, is 3 billion nucleotides, right? The thing is that this genomic data is only useful when it gets associated to phenotypic recordings. And as you know, these phenotypic recordings are actually being more and more common. We are all being recording ourselves, right, with these wearable devices. Soon we'll have implantable devices that will be collecting continuously data on the functioning of our body, on our physiology. And we actually maybe think that there will be medical nano-wearables, so machines that will travel through our body that will be collecting huge amount of information that, correlating with the molecular information, will in principle help to predict which is the status of our body, and to anticipate critical moments, right? So anyway, don't want to speculate on this. The second area in which will be a huge data generation is in actually characterizing the cells within the human body. Within the human body, there is 10 to the 13 cells. That's a huge amount of cells. It's billions of cells. And all my cells in my body have the same genes, have the same genome. But the cells in my body are very different. The cells of my brain are different. The cells of my skin, or the cells of my bones, the cells of my liver. Why is this? Even though they all have the same number of genes, 20,000 genes, that's the number of genes encoded into the human genome, genes that code for proteins. So the thing is that the thing that makes the cells different to each other is that not all genes, work on all cells in the same way. And with the aim of this project called the Human Cell Atlas is to measure the expression, how the genes, which is called expression, how the genes are expressed, what is the levels of the genes in each of these 10 to the 13 cells in the human genome. And you got to think that for each of these cells, we'll have 20,000 measures onto a sequence, which is 3 billion nucleotides. So the amount of data, it's really mind-boggling. This is just an example in which this is a plot of 50,000 cells, in each of which the expression of these 20,000 genes has been measured. And by plotting this into reduced by this dimensional reduction methods, I think this is TSNE. So we actually are able to identify the different types of cells that are into the human body, and actually new types of cells that we did not know were existing. The final project I want to mention is the Earth Biogenome Project. This is also one of these huge projects that aims to sequence the genome of all the eukaryotic species that are known to live on Earth, which is 1.5 million. I mean, we don't know how many species live on Earth. It's probably twice, 3, 4 times this number. Many of them are disappearing every day. And the aim of this project is actually to collect and sequence the genomes of all these species during the next 10 years. So all these projects will generate a huge amount of data that will generate many challenges for the field of informatics, both in terms of data storage and in terms of data analysis. But let me finish though with just suggesting that while biology is generating the problem, in biology may also be the solution, right? Because underlying this, what I believe, this very intimate relationship between biology and computation, and maybe in part the success of the term bioinformatics, is that actually biology can be used to compute. And actually, this is one of the first examples. This is the pioneer work of Leonard Adleman in the mid-90s, in which actually he tried to solve a problem, a typical problem in computer science, the tribal salesman problem, but actually using DNA both to code the problem and to find a solution using molecular tools. And actually he showed in the practice for a very small instance of the tribal salesman's problem that this problem can be solved using DNA as a computational instrument. But not only that, actually there is now an area I think in informatics, which is molecular computation. I'm not sure whether this is actually will have any practical application at all, but I think from the conceptual standpoint, that's actually very, very interesting, because it's the idea of biology as a computation. But actually, as a way of storing information, DNA may actually work, right? This is a paper published quite a few years ago already, in which actually these authors actually encode a number of information in a DNA sequence. And there was videos, there was Shakespeare text, there was some music, etc., etc. And actually, what they show at this time is that while recording is expensive because you need to sequence the DNA, actually, reading is not so expensive because reading means sequencing, and sequencing methods are quite inexpensive now. There is a projection of how long you need to store the data for storing in DNA be efficient, depending on how much expensive it is to synthesize DNA. But actually, for long-term storage, DNA may actually be a competitive way, mostly because we get high density of storage. That at that point, this author says that they claim that they could store 2 petabytes per gram of DNA. There is, I think, at this moment, no other way that is so efficient in storing data as the DNA. But actually, more recently, some other authors actually have devised instruments that are able to store data at really huge densities of 215 petabytes per gram of DNA. So let me finish with this. Thank you for your attention. Essentially, what I just wanted to say is that the data generated by biology has an intrinsic nature that makes it very amenable to computation. That's partially the success of bioinformatics. And actually that at the most basic level, the biological processes are also sort of computations. And that's why actually DNA can also be used as a computational engine. And again, thanks for listening to me. And thanks again for inviting me to this meeting.